Hello, everyone. In this chapter, we are going to go beyond Mendel's laws. Now, as a reminder, with Mendel's law, we looked at dihybrids and monohybrids. In monohybrids, we saw that genes can occur in alternate forms, right? And that these alternate forms were called alleles. You have some alleles that are dominant, others are recessive. And we did say that the two alleles are carried by homologous chromosomes, and that during meiosis, these chromosomes separate with each other to end up in different gametes. So with that, Mendel came up with the law of segregation. Then when looking at more than one feature, we were particularly in a case of dihybrid, Mendel showed that, well, when looking at two features, the two features may be carried by two separate chromosomes, and that at that point, you have the law of independent assortment. That was roughly Mendel's law, right? But we also started a discussion including the fact that, well, things are not that simple in human and other living organisms, right? There are more complex aspects, including the fact that some genes can affect the expression of other genes, and also the fact that the environment plays a very important role in expressions of genes and even the phenotypes that we see. So going beyond Mendel's law is going to cause us to dive into gene expression that appear to change Mendelian's ratios. We're going to take a look at lethal allele combinations, multiple genes, dominance and recessivity, and particularly in complete dominance and codominance. We're going to take a look at the role of the environment. We're going to take a look at how one gene can affect the expression of another gene. So we're going to talk about epistasy. We're going to talk about penetrance and expressivity. We'll talk about pleiotropy and phenocopy. And then we're going to address mitochondrial DNA as ways to affect inheritance. We're going to talk about mitochondrial genes, and also when genes appear on the same chromosomes. So our first step is going to be when gene expression appear to alter Mendelian's ratio. But in reality, even though they show to affect Mendelian's law, in reality, they are truly not, right? Now, just as a way of reminder, Mendel's stretch shows two distinct forms, dominant and recessive. But really, in real life, we see that it's not as much of a clear cut because some genes happen in more than two simple alternate forms, one being dominant and the other one recessive, right? You have in the population multiple alleles of the same gene. And these alleles are going to behave differently. We are going to see that a little later as we develop in this chapter. So most genes do not exhibit simple inheritance, a clear cut of dominance and recessiveness. No. We are going to see that in most cases, there are three general phenomena that seem to be exceptions to Mendel's law. But in reality, they are not exception. These are actually the rules, right? We're going to take a look at gene expression, mitochondrial inheritance, and linkage. In several circumstances, phenotypes ratios appear to contradict what we learned in the previous chapter with Mendel's law. But in reality, no, because this law are still going to apply regardless. Now, there are some alleles when in combination will never actually persist because they are simply lethal, right? A lethal genotype is going to cause death before the individual can reproduce. And actually, most of the time, these lethal genotypes are going to occur during embryonic stage and or during fetal stage of development, so that you are going to have almost systematic miscarriage, right? Double dose of dominant allele may be lethal. This is what we see in a case of achondroplasty, right? In dwarfism. So dwarfism, it's actually the case of gain of a function. Now, in a person with normal height, the feature is recessive. In a person with achondroplasia, the feature is dominant. It's a gain of function that occurs in a gene that is called FGFR3. Okay, so FGFR3 actually causes inhibition of growth in long bones, both in the lower limbs and in the upper limbs, right? By causing inhibition in the growth of long bones, the individual end up relatively short, not relatively, shorter than normal, right? That's what we call dwarfism, okay? So here we have the case of a movie star slash actor, Peter Dinklage. He received many awards, including for the Game of Thrones, right, where he received some important awards there. Now, this actor actually developed achondroplasia. Now, how does it show when someone develops achondroplasia? So basically, we are going to take a look at here the alleles that are responsible for the normal height. So lowercase a, it's normal height, right? That's the wild type allele, while dominant allele A, capital A, it's a gain of function that cause achondroplasia. Now, individuals that are monozygous, capital A, these individuals are not going to be able to survive. Right? This combination is lethal. While capital A, lowercase a, so heterozygous, these individuals are going to develop achondroplasia, therefore dwarfism, while lowercase a, these individuals are going to have normal height. So, in a cross of two individuals with achondroplasia, we are going to have the following ratios. So, achondroplasia, so capital A lowercase a and capital A lowercase a. So now, when we bring this capital A and this capital A, we are going to have an individual that is homozygous dominant. This homozygous dominant is going to die as embryo. So really, this doesn't count when we are looking at the ratios in the offsprings, because this is lethal, right? So we are going to focus on only survivors. So the chances of surviving are only 3, right? Of the 3, you are going to have 2 cases possible with achondroplasia, so that's 2/3, and 1 case normal height when crossing 2 individuals with achondroplasia. So really, we show here that you have 1 individual with normal height, 2 individual possible with achondroplasia, and 1 lethal. So we have a ratio to 1 to 1. And again, when we look at the offspring, we are only considering those offspring, those individuals who have a chance to live, so we don't take these guys into account. The dominant homozygous, we don't take them into account, right? So then, what would be the ratio if we cross a normal parent with a parent with achondroplasia? In this case, we are going to have 50% of chance to have a child with achondroplasia and 50% of chance to have a child with normal height. How so? Well, for this parent, the gametes are going to be necessarily lowercase a. While for this parent, the gametes are going to be capital A for one and lowercase a for the other, so that with capital A fertilizing this egg and this egg, we are going to have heterozygous here and heterozygous here. And these 2 individuals are going to develop achondroplasia, while the recessive allele here, when crossing other 2 recessive alleles, are going to produce individuals with homozygous recessive, which are normal. So we have 50-50, right? 50-50, right? So we are showing the same thing here in terms of ratio. So 1 chance of having an individual who is homozygous recessive, and 1 chance to have an individual who has heterozygous genotype, so dominant and recessive allele, which is 1-1, so 50-50. All right. In a case with multiple alleles, really individuals are going to carry 2 alleles from each autosomal gene or each autosomal chromosome, right? So 2 alleles. Now, when we look at the population though, there are multiple versions of alleles in the population, right? Which is actually going to cause things to not look so simple, so much of a clear cut, right? Gene can have multiple alleles because its sequence can deviate in many ways in individuals, right? consider all the people you know of, right? You have some friends, you have some cousins, you have people actually you just got acquainted with, right? You all have the same genes, but different alleles based on your genetic history, right? So that a particular gene is going to have multiple forms of alleles in a population. So different allele combinations can produce variation in the phenotype. That is very important aspect to remember. So an individual with two different mutant recessive allele for the same gene are going to be basically what we call a compound heterozygous, right? Because of the different forms of alleles that exist in the population. So some alleles, when expressed, are going to show what we call incomplete dominance. This is the case of familial hypercholesterolemia. Hypercholesterolemia is when you have too much cholesterol in the blood, right? Now, the cholesterol form we are considering here, it's actually the one that we call LDL. That stands for low density lipoprotein. Lipoprotein. Low density lipoprotein, LDL. That's the bad form of cholesterol. That LDL, to be imported in, requires a presence of receptors embedded in our plasma membranes, right? Unless we have this receptor embedded in our plasma membrane, we cannot internalize this LDL. So in the population, most individuals are going to have the receptors on the surface of their cells so they can internalize those LDL so that we have a normal amount of LDL in the blood, right? Typically low level of LDL in the blood. So in individuals, in the general population, we're going to have low cholesterol in the blood. However, individuals that actually have one allele that does not express are going to only have half the normal amount of receptors on the surface. So here for heterozygous individuals, again, that express half the number of receptors of cholesterol, we are going to have a high level of cholesterol in the blood. Now, in individuals who are homozygous, these individuals are not going to have receptors on the surface of their cells. Therefore, we're going to have a high level of cholesterol in the blood, which actually is going to cause the death of individual in a relatively early age of their lives, right? They're going to have stroke. They're going to have many kind of heart cardiovascular problems because of those high level of cholesterol. They're going to have plaques. They're going to have atherosclerosis in our blood, in their blood vessels. Then we have the case of codominance. This is typically what we see in the case of ABO blood type. Now, in the case of ABO blood type, we have three alleles that exist. We have one that is IA, the other one is IB, capital I should say, and the other one is lowercase I. Now, an individual who is type blood type A, that individual is actually going to have an enzyme that causes the production of a protein with a sugar attached to that protein, causing an antigen cause A to be present on the surface of the red blood cells, right? This individual with blood type A have two possible genotypes. One genotype is capital IA and capital IA. That's one possibility of genotype. The other possibility of genotype is capital IA and lowercase I. These are the two possibilities for the individuals who are blood type A. For individuals who are blood type B, you have a variant of the enzyme that this time, instead of producing antigen A, they are going to produce antigen B. So these individuals are producing a different form of enzymes. That enzyme produce antigen B on the surface of red blood cells. Now, for genotypes, individuals are going to have two possibilities. They can be capital IB and capital IB, or they can be capital IB and lowercase I. How about individuals who are blood type AB? Individuals who are blood type AB are actually going to produce the two enzymes, the two form of enzyme. The enzyme that produce antigen A and the enzyme that produce antigen B, so that on the surface of the red blood cells, we are going to have both antigen A and antigen B that are going to develop. Now, in this case, only one possibility of genotype, capital IA and capital IB. How about individuals that are type O blood? Well, those type O blood are going to have one single possibility. It's lowercase I. These individuals do not express any antigen. And again, the antigen here is going to be the sugar that is attached to the protein, right? In A, it's one kind of sugar that is an antigen. In B, it's a different kind of sugar that is an antigen, right? So type O does not have any antigen A or antigen B. Now, specifically, how do you show what offsprings can be obtained from individual cross? So say we have one parent who is type A and one parent who is type B, right? So considering the two possibilities, right? Considering the two possibilities of genotype between A and B, well, we are going to have, in this case, capital IA, capital IA, capital IB, capital IB. So when we look at that, we see that all individuals are AB in this particular instance of a cross between type A and type B. Now, if a parent A is capital IA, lowercase I, while the parent type B is capital IB, capital IB, we are going to see that, well, you have 50% of chance of having children who are AB and 50% of chance to have children who are B. Notice that there is no child here who is only A, right? Now, another possibility of cross here, type A with capital IA and capital IA here, and the B, capital IB and lowercase b. Here we have 50% of chance of having children AB and 50% of chance to have children who are A. Now, how about we cross a type A who is heterozygous, right? Capital IA and capital I mean lowercase I, I'm sorry. And a type B that is also heterozygous, so we have capital IB and lowercase I. In this particular case, there are multiple possibilities for phenotype. Chances of having individual AB is 1 out of 4. Chances of having individual B is 1 out of 4. For individual A, it's 1 out of 4. And now see that we do have a chance to have a child with type blood O, right? That is 1 out of 4. So sometimes we think that whenever we are a particular blood type, that a child has necessarily to match either your blood type or the blood type of the other parent. But in reality, no. A child can have a blood type that does not match any of the parents. That doesn't mean that that child is not the child of both parents, right? So we have to be cautious with drawing quick conclusions, right, as to who is the father of a particular child. All right, let's take a look at epistacy. What is epistacy? Epistacy is actually a phenomenon where one gene is going to affect the expression of another gene that is completely different. When I say completely different, we are not talking about alternate forms of the same genes here, no. We are talking about one gene, say gene A, another gene, gene B. So gene A can actually, when expressed, affect the expression of gene B. Or actually, when gene A is absent, that can affect the expression of gene B, right? So everything depends on the interactions that are happening in the cell. So let's take the look of a blood type that we call the Bombay blood type or Bombay phenotype. In the Bombay phenotype, you have a particular gene that is the H gene. The H gene is actually epistatic to the I gene. What we say, it's that what we mean, it's that the H gene affect the expression of the I gene. How so? The H protein is able to place a molecule on the surface of cells with A or B antigens. Let me repeat that. The H protein places a molecule at the surface of blood type A or blood type B. Now, homozygous recessive lowercase h, right? Genotype, they do not produce the H protein. So, as consequence of that, without the H protein, the A or blood antigens cannot be attached to the surface of red blood cells. So mind that an individual is still capital IA or capital IB, but without the protein H, the B is not going to be attached to the surface, or the H is not going to be attached to the surface. As consequence of that, the individual is actually going to develop a type that doesn't match its genotype. Let me repeat that. The individual is going to express a phenotype that does not match its genotype, because that genotype is being affected by the lack of expression of another completely different gene. So all lowercase h genotype, right, have the phenotype of blood type O, even though they may have capital IB slash capital IB or capital IA, right? Or even in the case of capital IB and capital IB, right? So individual capital IB, capital IA should be AB. But in the case of homozygous recessive H, this individual will never be AB. They will be O, right? So that's what we call the Bombay phenotype. When one gene is affecting the expression of another gene. So H gene is epistatic to the I gene. Then we are going to dive into something that is a little more complex here: the penetrance in expressivity. Now, these two terms are going to describe the degree of expression of a single gene. The rats that you see here, they have the exact same genotype. They have the exact same genotype, but their phenotype is not the same. Look at the fur color. The fur color is different in these three rats. Well, this is when penetrance and expressivity comes into play. What do we call penetrance? Penetrance, it's basically a all or none expression of a single gene. Question: the penetrance, when we address the penetrance, we are asking, is that gene being expressed, yes or no? Right? That's the question with penetrance. For expressivity, the question is, what is the extent to which that gene is being expressed? Yes, the gene is being expressed, but to which extent? Right? When we consider a population, right, what is the proportion of a population that expresses the gene? What is the proportion that does not express the gene? Right? So really, the story is more complex here. So one, is the gene being expressed, yes or no? Two, what is the severity of the expression, or what is the extent to which the gene is being expressed, right? So this causes some funny phenotypes, right? So that the genotype can actually have what we call incomplete penetrance. Incomplete penetrance means that, well, there is no really clear cut as to, is the gene being expressed, yes or no. Well, the gene can be expressed, but with a certain severity, or to some extent, or to a lesser extent, right? Okay? So we talk about incomplete penetrance when some individuals do not express the phenotype, right? They do not express the phenotype. So the phenotype is going to be expressed with a certain variation in a population based on intensity of gene expression. This is what we show down here, where these three individuals have the exact same genotype, but their phenotype is different, right? So you see that when it comes to human genetics, things are not always a clear cut, right? Then the other aspect we are going to address here that tend to alter Mendelian ratio, but really do not alter Mendelian ratio, is the case of pleiotropy. In pleiotropy, this is the case where one particular gene is going to have multiple effects, right? So one gene is going to control several functions in a person, or has more than one effect. This is true for cystic fibrosis, for instance, right? In a cystic fibrosis individual, right, some individuals can have some affection in the lungs. They can have affection in their digestive tract. They can have affection in their reproductive tract, and so on. So this is pleiotropic, right? So the pleiotropy actually can be difficult to trace through family. Why? Because people with different subset of symptoms may appear to have different diseases. For instance, someone with cystic fibrosis, some individual with cystic fibrosis can have infertility issue, while other people with cystic fibrosis do not have infertility issue, but they have chronic respiratory issues. Other people with cystic fibrosis may not have chronic respiratory issues and not have fertility issues, but have chronic digestive issue, right? So this is one good example of where it would be difficult to trace things, right? And associate these different problems with one single gene, which is the gene for cystic fibrosis, right? Another example is the Marfan syndrome. The Marfan syndrome, it's basically a genetic defect where there is a misproduction of a particular protein of the connective tissue. That protein is called fibrillin, right? Fibrillin is actually elastic, right? So that fibrillin, when it is not properly produced, can cause different phenotypes, including lens dislocation, the lens in your eye, right? Lens dislocation, unusual long limbs, long, very long fingers, thin and long, right? And in the chest is going to be caved in. Now, not everyone with Marfan syndrome has a chest that is caved in. Okay? Not everyone has extremely long limbs, right? You're going to have a variation of these effects in individuals, right? The most serious symptoms of the Marfan syndrome, it's actually going to be when the aorta is going to be weak, right? Because of the lack of fibrillin, right? That aorta can actually burst. And when it bursts, this can cause a sudden death of the individual. And then we have genetic heterogeneity. In genetic heterogeneity, we're going to have that different genes can produce identical phenotypes. Now, here, you know that genes may encode enzymes, and these enzymes are going to catalyze the same, be part of the same metabolic pathway. Let me repeat that. Genes may encode enzymes that are part of the same metabolic pathway, right? Or we can have different proteins that are part of the same pathway. Okay? Different proteins are part of the same pathway. So, for instance, in a case of osteogenesis imperfecta, this is when the bone formation is not proper, so the bone doesn't have the proper density, right? The density is relatively low in osteogenesis imperfecta, and actually, as the person grows in age, that osteogenesis imperfecta can lead to osteoporosis, right? Which is when your bones are brittle and break easily, right? At least 8 genetically distinct forms of osteogenesis imperfecta are recognized in the population. At the end of the day, these different genetic type are going to lead to the exact same phenotype, which is not osteoporosis, but osteogenesis imperfecta, which is a low form of osteoporosis, really. And then we have the case of retina dystrophy that can lead to blindness, right, to individuals. So we have multiple genes that are involved in this, right? So in reality, you have multiple ways to become blind at some point in your life, right? You're going to have, for instance, the rods that can be affected, you have a particular number of genes that, when altered, can cause blindness. That's for the case of rods, right? In the case of cones, you have some other sets of genes that, when affected, can lead to blindness. So not one single gene is responsible for this, but multiple genes. And the the sequence of expression, or the onset of gene expression or lack of expression, is also going to affect when this phenotype blind is appearing in someone's life. One can start losing sight in their early age, while other people can start losing a sight in early adulthood or late adulthood, right? Then we have the phenomenon of phenocopy. What is phenocopy? This is case of traits that appears inherited, but in reality they are caused by the environment. Okay? So we are going to see some symptoms that look like they are being passed down from one generation to another, but really the trait appears because of environmental factors. That's, for instance, the case of exposures to teratogens. And teratogens, remember, are these factors that are rather environmental, that affect the development of an embryo slash a fetus. Everything depends on the stage, right? Thalidomide were shown to cause development of limbs in an early stage of development, right? So they cause defect in limbs, right? Either short arms or short legs, or both short arm and short legs, right? That appear in an individual. This actually effect of teratogens, in this case, thalidomide, right, is going to behave as if this was a genetic defect, which actually does occur, the genetic defect, right? Other case is the case of AIDS or HIV that is passed down from mom to the baby. So when mom gives birth to a baby with HIV infection, we may think that, oh, that's a genetic issue, but actually it is not, right? It is completely environmental, right? There is exchange of fluid between mom and the baby, right? When the baby is still in the womb, right? Some factors, including viruses, are able to cross the placenta barrier, right, and find themselves in the baby, and at that point the baby is HIV positive, right? So that's the case of phenocopy. And now let's address genome sequences. So genome sequences show a number of things, including the fact that human genetics, as portrayed by Mendelian genetics, is not as simple as it appears, right? Some features were thought to be simply the caused by one particular gene that is defective, but we see that things are more complex than that, right? Terms like epistacy and genetic heterogeneity are terms that were not known when looking particularly and exclusively at Mendelian genetics, right? Okay? Morphan syndrome. It's one particular case that that the Mendelian genetics could not explain, right? Interactions between genes are showing different levels of penetrance, different levels of expressivity, things that actually we could not comprehend with the sole Mendelian genetics, right? Some examples are the Huntington disease that exhibit different forms in a population and cannot be explained by the simple by the simple way of Mendelian genetics. So this slide is actually a summary slide of different ways that Mendelian genetics seems to be altered, but that is not actually altered. We just show that things are more complicated than they are, right? You have, for instance, the phenomenon of lethal alleles, right? The effect, it's that homozygous dominant is simply lethal, right? Achondroplasia, it's an example of that, right? Multiple allele, so many variants or degrees of phenotypes are possible. We have an example, cystic fibrosis. So please review this entire slide. It's going to be very relevant, very important, right? And going to help you better grasp the information. Now, let's move to the next stop in terms of learning objective for this chapter, the mitochondrial genes. The mitochondria in our cells are responsible for the production of much of the ATP we need for cellular work, right? That ATP is produced through cellular respiration, right? And one particular stage of cellular respiration is the electron transport chain. That electron transport chain is what that produce the most ATPs. You know that during fertilization, a sperm cell is going to fertilize an egg. An egg is exceptionally huge compared to the sperm cell. How is that possible? This is possible because during meiosis, the production of egg cells, we have unequal distribution of the cytoplasm among daughter cells, so that we have polar bodies that are very teeny tiny, while the oocyte is huge, inheriting most of the cytoplasm, bunch of mitochondria and all other cytoplasmic organelles, right? The sperm, on the other hand, when it's coming for fertilization, really brings one very important thing. It's the nucleus with its DNA content. It doesn't bring anything else. right? So you have that in individuals, all their chromosome, all their mitochondria are basically coming from mom, right? So for instance, in here, in this particular pedigree here, when we look at the offsprings, so dad, obviously, dad's mitochondria come from his mom, right? Mom here is going to have mitochondria with a particular feature that we are going to follow, right? We're going to see that mom is going to transfer that feature to all their offsprings, their offspring. Okay? Everyone is receiving that feature because of maternal mitochondrial DNA, right? When we look in the second generation, the males that receive the defect, the mitochondrial defect from mom here, did not transfer their defect to their children, right? While the mother here is transferring the defect to all their children down here, the dad here, no children is affected. The mom here, all children are affected. All right? So this is to show that mitochondrial DNA, it's a matter of transmission from mom to offspring. Dad doesn't transmit any mitochondrial DNA to offspring, right? So there are few characteristics about mitochondrial DNA. What are those characteristics? One is that mitochondrial DNA, right, do not experience crossing over, which means there is no possibility of recombinations of DNA in there. That's one. Two, it's that mitochondrial DNA mutates faster than the DNA that is found in the nucleus. Why? Because the DNA repair machinery in the mitochondrion is not as effective as the DNA repair machinery in the nucleus. That's one. Two, it's that the mitochondrion, it's where oxygen is going to be used. Oxygen is very damaging, right? Oxygen produce free radicals. And these free radicals have to be discarded, right? But the mitochondrion is not as effective at discarding these issues caused by the free radicals. Therefore, we are going to see some defects in the DNA. So mitochondrial genes are not wrapped with protein. We saw that in the nucleus, DNA is combined with some proteins called histones, and that together they are going to form a compact structure that is going to protect DNA. But here, the mitochondrial genome does not have histones. Therefore, it's going to be much more vulnerable to attack from environmental factors. Mitochondrial genes are not interrupted. In other words, in the mitochondrial genome, you don't have exons and introns. Okay, let me repeat that. You don't have exons and introns. Introns are non-coding part of the chromosome, while exons are coding part of the chromosome, right? So all the DNA encode for something in the mitochondrial genes. And mitochondria with different alleles from the same gene can reside in the same cell, remember that the DNA machinery repair is not effective in the mitochondria as it is in the nucleus. Therefore, you can have some cells with mitochondria that are different genetically. Yes, that can happen, right? So, some mitochondrial disorders, some mitochondrial disorders can be passed down, but only from mother to children, right? Mitochondrial genes are going to encode proteins that are involved in protein synthesis. Now, proteins that are being synthesized in the mitochondrion. That's very important to note, right? Two, the mitochondrial genes are actually going to be involved in energy production. So two types of genes in among mitochondrial genes. You have some genes that are involved in protein synthesis and some genes that are involved in energy production. So several diseases are going to result from the mutation of mitochondrial gene. Some examples are going to be mitochondrial myopathy. The mitochondrial myopathies are going to, myo means muscle. Okay, myo means muscle. Pathy means not properly functional, right? Myo, muscle, pathy, not properly functional, dysfunctional, if you will. So there are some cases of inherited diseases in which children are going to be born with weak muscles, right? Muscles are weak. They are flaccid because the mitochondria are not producing the energy that is needed to sustain the tonicity of the muscles. Another example of mitochondrial disease is the liver optical atrophy, where the individual is going to have an impaired vision, right? Now, theoretically, a woman with mitochondrial disease can avoid transmitting it to their children, but how? Only if her mitochondria can be replaced with healthy mitochondria from a donor person, right? That's theoretical. But what is the chance? What is the chance that in an egg we can replace all defective mitochondrial DNA, right? Technically, we can extract the mitochondria and replace them with the normal one, but that's in theory, right? That's in theory. Okay, let's take a look at heteroplasty. Heteroplasty is a condition where mitochondrial DNA sequence is not the same in all copies of the genome. And again, I'm going back to the DNA repair machinery in the mitochondria is not as effective as in the nucleus, right? So that the genome can be different in the same cell, right? In the same cell, the mitochondrial genome can be different, which causes genotype at the mitochondrial level to be different. Therefore, we may see a difference in the genotype too. So this is called heteroplasmy, right? So mitochondrial DNA are going to exhibit different alleles for the same gene in the same cell, right? At each cell division, we are going to see that because of cytokinesis, well, there is no prediction whatsoever as to how the distribution of mitochondria is going to occur. So daughter cells can inherit different amount of mitochondria and therefore exhibit not only a different genotype but also a different phenotype, right? If an oocyte is heteroplasmic, different number of copies of the mutant mitochondrial DNA may be transmitted to the gametes, therefore to the offsprings, right? So the proportion is going to differ from one individual to another individual. So here we are showing an egg with different type of mitochondria, right? So this mitochondrion right here is going to have one particular genotype, and this one is going to have a different genotype. So the dark one is mutated while the yellow one is normal, right? So let's see how this can affect the offspring. So upon fertilization of this egg, right, we are going to see something happening during the development. So we can be in this situation here with a wild type phenotype where the wild type mitochondria are much more abundant than the mutated mitochondrial DNA. So we're going to have a wild type here. And then there is a certain threshold beyond which the cells are not going to exhibit the wild type phenotype, depending on actually the number of copies of the mutated. So we have here 4, more than 4. So we have 5 and more, where we are going to have the phenotype of mutation that is going to be expressed. But interestingly, the phenotype of mutation is going to be expressed with a different level of severity. Okay? With the low copies here being less severe than the most copies here. Okay? And this is what we call heteroplasmy. Now, finally but not least, let's look at the linked genes. The linked genes can appear on the same chromosome. So here we are going to show the work that William Bateson and Punnett did. So Bateson and Punnett worked on pea plants, just like Mendel, but they encountered some issues pertaining to Mendel's law. In this particular case, we are going to take a look at plants with purple flowers and long pollen, true breeding. So purple flowers being with a genotype capital P, and long pollen grains being with a genotype capital L. This is a true breeding for purple flowers and long pollen grains, and another true breeding with red flowers, with round pollen, with lowercase p, homozygous recessive for red flowers, and homozygous recessive for round pollen, lowercase l. So using these two plants as true breeding, Bateson and Pennant were able to actually find some interesting things. So, assuming that here the chromosome has these two alleles, these two genes on the same side, and these two genes on the same side, right? This is one form of allele here and another form of allele here. During meiosis 1, this is what we got, right? During meiosis 2, we have that every single gamete has a parent genotype, parental genotype here with capital P, capital L. So this one here will be capital P, capital L, and another capital P, capital L for this one here, right? Then we have lowercase p, lowercase l for this one here. And finally, lowercase p, lowercase l, this one here. So that's basically the simplest form of anticipation of what gametes would be produced. But we know that during meiosis, things are not that simple, because during meiosis, and specifically during prophase 1, there is a particular event that occurs that is a crossing over, where some exchange of genetic information can occur between homologous chromosomes, right? Between non-sister chromatids, right? Between non-sister chromatids. In other words, between this leg and this leg, we can see a crossing over developing, all right? And that is actually going to lead to a number of interesting features, okay? Now, before we dive into those features, let's take a look at the hybrid prediction for Mendelian genetics. In Mendelian genetics, when you cell cross a D hybrid here for purple flowers and long pollen grains, technically doing that the hybrid cross, you would have these gametes, right, for one parent, and these gametes here for another parent, and the phenotypic ratios would be 9-3-3-1. That is the law of segregation, the law of independent assortment, right? Outcome, okay? So that's the prediction. But Bateson and Punnett found something completely different. They observed something completely different. They observed a ratio 3-to-1 phenotypic ratio. That was confusing, right? They're like, okay, well, what is happening here? And this is when they came with the idea that, wait, something must be happening here that we did not anticipate. What is that something? That something is DNA recombination that occur during crossing over between homologous chromosomes, right? So chromosomes recombine during crossing over in prophase 1 of meiosis. I want you to remember that. So these new recombinations are going to cause some chances of gametes that do not look like either of the parents, but it's actually a mix of both parents, so that at the end of the day, one parent is a dihybrid parent is going to produce some parental chromosomes and recombinant chromosomes, right? And this happens during the process of crossing over, where two non-sister chromatids, let me repeat that, non-sister chromatids, right, are crossing their legs. And as the non-sister chromatids are crossing their legs, when that crossing is effective and the exchange is effective, what happens is that now we're going to have a recombinant leg here and a recombinant leg here. At the end of meiosis 2, what do we have? We have a parent gamete here, another parent gamete here, but a recombinant gamete here and another recombinant gamete here. All right? So that we are going to ask, wait, for a dihybrid, what is the proportion of recombinant in the population, and what is the proportion of parent phenotype in the population? And now we are going to dive in this complex aspect. So now, to figure that out, we are going to take a look at how how genes appear on chromosomes. This gene can appear on chromosomes in two manners. One is that one chromosome here that you receive from dad has the gene here with dominant allele and the gene here with dominant allele. So here for purple flower and long pollen grain. From mom, the allele recessive here for red flowers, and the recessive allele here for round pollen grain. So these two dominant alleles for those genes are going to be carried by the same chromosome. The recessive alleles here for the different genes are going to be carried by the same chromosome. That's one organization. But then another configuration, it's that the dominant allele of this gene here can be on the same chromosome as the recessive allele from the other gene. And same thing over here. That kind of organization is called transorganization. All right? It's called transorganization. So that trans configuration is very important when it comes to what could be the outcome in terms of gamete formation at the end of the day. So here, the frequency of recombination is going to be based on how far apart the genes are going to be on the same chromosome. So while A and B are on the same chromosome, they are very far apart, right? While B and C are very going to be close together. Now, we can actually determine how far apart A and B are, and B and C are, and A to C are, right? This is going to be very important to actually be able to map the location of the genes on the chromosomes, one in relation to another. That work was done by Thomas Hunt Morgan. Thomas Hunt Morgan was able to come up with a correlation between the frequency of recombination and the distance of genes on the same chromosomes when linked, right? So Morgan came with the idea that when genes are far apart, say gene A and B are far apart, these genes are more likely to be involved in crossing over compared to genes that are so close together. The closer the genes are together, the less likely the chance that they would be involved in crossing over. Therefore, these two genes that are so close together are likely to end up in the same gamete compared to these two genes that are far apart together that are very likely to end up in two separate gametes because of occurrence of crossing overs, right? All right, so basically, this is what we show here. When genes are linked, right, with a cis organization, right, what we are going to see is that the two genes being far apart after meiosis, actually, we are going to have here parental allele configuration here and recombinant allele configuration here that actually may approach 50% of recombination. And this may approach 50% of the recombination by virtue of being so far apart, right? In other words, beyond a certain distance, as they are far apart, the genes are going to behave as if they were on separate chromosomes. Let me repeat that. There is a certain distance when genes are far apart that even though they are on the same chromosome, they are going to behave at the end of the day as if they were on different chromosomes, leading to the idea of actually that we cannot always tell in independent assortment if the genes are carried by different chromosomes or they are carried by the same chromosome because of the distance at which these two genes stand apart from each other on the the chromosomes. Okay, these are details that are very important to remember. Now, because of the work of Morgan, Morgan came with the idea that we can actually align genes on a chromosome based on the proportion of recombination, right? So he said he came up with the idea that 1% of recombination equals one map unit, and that one map unit is 1 centimorgan. Let me repeat that. 1% of recombination is one map unit, and that one map unit is 1%, 1 centimorgan. I'm sorry, 1 centimorgan. In other words, if we have two genes X and Y that are 10 unit apart, that 10 unit apart, it's actually centimorgan, 10 centimorgan, right? The distance between X and Z, it's 4 unit. 4 unit, it's 4 centimorgan. But 4 unit, it's also 4% of recombination. 10 unit, it's also 10% recombination. All right? Now, knowing the distance between X and Z and Y, between X and Z, between Z and Y, now we can tell what the distance is, right? No, not what the distance is, but how the genes are organized on the chromosome. So X and Y, the distance is 10, as you can see here, right? Between X and Z, the distance is 4. Between Z and Y, the distance is 6, right? So that Z sits between X right here and Y right here. All right? So looking at this is very important, because if we are not cautious, we may say, for instance, that Z is located right here, which Z could very well be, right? Because the distance between X and Z is 4. But if we put Z right here and putting X right here, this would mean that the distance between Z and Y would not be 6, but it would be 14, which is not true, because that's not what we see here, right? Okay, so this is a way to kind of like show you how to map out genes on a chromosomes. Now, in the linkage, there is a particular aspect that we call linkage disequilibrium. The linkage disequilibrium developed from the non-random association that exists between DNA sequences. Let me repeat that. Non-random association between DNA sequences. In other words, when two genes are closely associated together, the likelihood that they end up in the same gamete is very predictable, right? It's very possible, right? The farther they are apart, the more they are likely to end up in separate gametes. And if they are so far apart, then they are very likely to actually end up in separate gamete too, right? In terms of independent assortment, right? So inherited together in the case of linkage disequilibrium, so genes that are closely together have a high chance to end up in the same gamete, right? Human genome is going to exhibit a lot of linkage disequilibrium. Why? Because genes are actually organized by block, right? They're going to be organized by blocks so that they tend to stick together more often than actually what we can predict, right? So, what we could predict earlier, right? Now we do see that we do have many genes that systematically end up together in the same gamete as other genes because of this LD blocks, right? These LD blocks are actually called haplotypes. Okay, haplotypes, it's basically chunks of or groups of genes that systematically end up in the same gamete because they are so close together, right? And knowing that haplotype, we can actually track the genes in a population and come up with ways to actually connect individuals and see migration of populations among human. Now, how do we solve how do we solve linked gene in a population? One example that we are going to take here is the example of the Neal-Patella syndrome. The Neal-Patella syndrome is actually a rare autosomal dominant disease. That autosomal dominant disease causes underdevelopment of nails, painful arthritis in the knees and elbows. Now, the genes for Neal-Patella syndrome and the ABO type, specifically the I, capital I, are going to be 10 map unit apart. And these two genes are carried on chromosome 9. Now, 10 map unit, it's also 10 centimorgan, right? 10 centimorgan. So, now two parents, Greg and Susan, they each have Neal-Patella syndrome. Now, Greg has type A blood, while Susan has type B blood. Okay? The question here is, what is the probability that their children has normal nail and knee and type O blood? Let me repeat the question. What is the probability that their children has normal nail and knee and type O blood? Now, remember the different genotypes that one can have when you are A and when you are B, right? You also know that Neal-Patella syndrome, it's a dominant disease, right? So whenever you have capital N, you express the disease, right? If we have lowercase n, then you don't have the disease. Now, provided that you are homozygous recessive, right? So, specifically here, we have a phenotype, Neal-Patella syndrome, right? For Greg, for Suzanne, Neal-Patella syndrome, but for blood type, Greg is type A, Susan is type B. In terms of genotype, what could be the genotype? Now, capital N, lowercase n, for Neal-Patella, capital IA, lowercase I, for type blood A, and for type blood B, we're going to have capital I, B. We don't know here if it's lowercase I or capital IB. Now, what are the possible allele configuration? So we have the possible allele configuration right here. Now, Greg's, what are the possibility based on the configuration, the different types of sperms that can be produced? So we have the sperms right here. So capital N, capital IA, lowercase n, lowercase I, capital N, lowercase I, lowercase n, capital IA. That's for Greg. For Susan, capital N, lowercase I, lowercase n, capital IB, capital N, capital IB, lowercase n, lowercase I. Remember, we want the child to be normal nails, normal knees, but O, right? In other words, we want the child to be lowercase n, lowercase n, lowercase I. What is the chance? So here are the frequency for the parents. 45%. Here, 45% here. The recombinant, 5% here, 5% here. So question, what is the chance that a child is going to be lowercase n, lowercase n? So that chance is going to be, now, not just lowercase n, but also lowercase I. So we see that right here, this gamete has to fertilize this gamete right here. That's what needs to happen. So now, knowing that this gamete has 45% of chances of developing, and this gamete here has a 5% chance of developing, so now we are 0.45 times 0.45. This is equals 0.025 or 2.25% chance of having a child with normal nails, normal knees, and blood type O. All right? So this is how this problem would be solved. Thank you for your attention. We are going to stop our discussion here.